All right. Good afternoon.
Good afternoon.
I like to hear people. I give you some love, you give me back some love. I like that. So I kind of like an interactive session. I mean, so let me tell you what I'm going to do. I'm going to tell you guys about my research, and then I'll tell you a little bit about how I got to where I am. And I hope that by the time we get to that point, you would be more comfortable with interacting with me and asking questions as we go. So I'm an associate professor in the Department of Microbiology and Cell Science. I've been there for quite a while. And so if you transfer into that department, I teach parasitology, human parasitology. And I also teach some advanced cell biology courses. So with the hurricane that has come through, we all are thinking about our homes, how our homes are built. And the interest that I have is really learning about how organisms that live in cells, how they develop, how they build the niche that they live in. So I study an intracellular parasite. It lives inside a host. And I'm very much interested in how that parasite develops the home that it resides in. And we call that home the parasitophorous vacuole of the parasite. So I'm interested in how the parasitophorous vacuole is formed. And as you'll see, the more we learn about how the parasitophorous vacuole gets formed, or the way a parasite builds its home, we can actually start coming up with strategies to disrupt it. And those strategies can then lead to the development of drugs. So I kind of want to give you a bit of insight into what we do in my lab. It's going to be one of the projects in my lab. If I go too deep or I lose you, please make sure that you stop me and ask a question so that I can explain things a little bit better. So the parasite that I work on is Leishmania, and I know that most of you might never have heard of the parasite Leishmania. So I'll tell you a little bit about Leishmania before I get into the biology of the parasite. So, oops, okay. So here are some images of individuals who are infected with Leishmania parasites. The disease presentation is very varied. So shown here are a couple of examples of, this is what's called cutaneous leishmaniasis. So you have a sore, or what we call a lesion, that develops at a site, usually where the parasite is introduced into a host. So this is a lesion. It's a cutaneous lesion. These are cutaneous lesions, but in this particular case, this is what we call disseminated cutaneous lesions. So these are disseminated cutaneous leishmaniasis. You usually start off with a primary lesion and then secondary lesions. In this particular case, these individuals, this kind of presentation starts off with a cutaneous lesion that then spreads into the mucosal areas. And this is an example of mucocutaneous leishmaniasis. So you have different kinds of cutaneous presentations, and then you have visceral presentation. Here, the parasite starts off at a particular site in a host, and it goes into the spleen and the liver. And what you get is very distended spleen and liver. And this kind of disease presentation, the host can die if they're left untreated. So the entire range of leishmaniasis, or the entire disease presentation range, is cutaneous, disseminated cutaneous, mucocutaneous, and then visceral leishmaniasis. So each of those different kinds of presentations can be seen in endemic areas, and I'll tell you a little bit about where those endemic areas are. In all, there are about 12 million to 15 million people who are infected with Leishmaniasis across the world. So it's a parasite that's significant in its impact on different communities. So the countries that are infected, so I'm sure you cannot read this from the back. For cutaneous Leishmaniasis, you have the Americas, so in Brazil, you can see some in Argentina, in Central America. These are all places that have cutaneous Leishmaniasis. In Northern Africa, in the Middle East, these are all places where you see cutaneous Leishmaniasis. As far as visceral Leishmaniasis, again, in South America, Brazil is a hotbed of a very endemic area. In northern South America, and again in Central America, all the way through in Europe, in places like Spain, Italy, you have visceral Leishmaniasis. And all of these things became even much more severe when people started coming down with HIV infections. So in places like Spain, in France, in Italy, these parasites infect humans, they also infect dogs. So there are places in parts of southern Italy where when you go, there are lots of dogs that are infected with Leishmaniasis. The highest concentration of Leishmania infections ends up being in India, especially visceral Leishmaniasis. And so there, you're going to have a lot of people who are going to succumb to the infection. So as you can see, throughout the tropics in most of the world and even beyond, Leishmania is a problem or is a significant concern. All right? So this is a parasite that's out there, and I should mention that in Florida, so there's Leishmania transmission in the US as well. In the US, there are dogs, kennels of dogs in northern New York that have been found with Leishmaniasis. It's not known how those dogs acquire the infection. In Florida, in Ocala, there have been 2 horses that have been detected with Leishmania infections. It's not known how those horses acquire the infection. So it's a parasite that's out there, even though you might not have heard of it, but it's a parasite that has a significant effect on human populations. So let me tell you a little bit about how the parasite is transmitted and how you acquire it. So as parasitologists, one of the things that we like to show is the life cycle of the parasite. Leishmania is transmitted by sandflies. And sandflies are these little bugs that are even sometimes smaller than mosquitoes. When sandflies go for a blood meal, and if the sandfly is infected, the morphological form of the parasite that is transmitted to a host is called a promastigote. These parasites have a flagellum. So when the parasite goes for a blood meal, and you know that, when this vector goes for a blood meal, you know that sandflies and other vectors usually go for a blood meal because blood helps them in the development of their own eggs. So they go for a blood meal, and the vector deposits the promastigote form of the parasite into a host. So in some hosts, depending on the parasite species, the point at which that sandfly goes for its blood meal is where you get those cutaneous lesions. So if the sandfly bites you on your hand, that's where you get the cutaneous lesion. If it bites you in your face, and on your face, that's where you get the cutaneous lesion. So that's where you get parasites introduced into a host. These parasites have to enter host cells. So these parasites cannot live free in the blood of a host. They have to enter host cells. So these parasites are intracellular parasites. So at the site of infection, it's kind of an interesting relationship that occurs. The sandfly, or when the sandfly bites, there's like sandfly saliva that induces cells to be recruited to the site of infection. And when those cells are recruited to the site of infection, the parasite enters the cells when they come to that site of infection. And the major cells that are infected are macrophages, neutrophils. So once the parasite goes in, the parasite lives inside cells. So this is a good example of what an intracellular parasite looks like. It lives inside the cell. So for us, those of us who are interested in how a parasite lives in a host or how a pathogen lives in a host, we are interested in all the steps. First of all, how it survives in blood. Next, how it gets into a cell. How does it know which cell to get into? There are lots of cells. How does it know which cell is a macrophage, which cell is a neutrophil, which cell is an endothelial cell? What will happen if it gets into cells that are not the right ones? Well, in the case of Leishmania, it mostly goes into cells in part because it's a bigger parasite and it needs a cell that can engulf it through phagocytosis. So once the parasite goes into the cell, it divides inside the cell, and eventually the cell is going to rupture, and the parasite will get out of that and infect other cells that are at the site. So the cycle will continue, and that's what leads to these lesions. So if we go back now and look at these lesions, essentially it's cells that are recruited to the site, the parasite infects them, more cells come, the parasite infects more, and you end up with, so there is a cycle of parasite death and parasite recruitment, which ends up resulting in these particular lesions. So just continue with the life cycle here. If a sandfly were to come in again and ingest blood from this host, it will acquire parasites, and those parasites will go on into the sandfly, divide into the sandfly, transform into promastigotes, and then essentially the cycle will continue. So the parasites, they spend time in a mammalian host, and once another sandfly vector comes by, it takes up the parasites transform and divide in that sandfly vector, and that sandfly can go on and infect someone else, right? So that's pretty much the way vectors will transmit. They acquire the infection, something happens, there's a period of division inside the vector, and when the vector goes back for a blood meal, it transmits the parasite. All right, so that's the life cycle. What I showed you is that for some parasites, those parasites stay at the site of infection and they cause cutaneous leishmaniasis. For other parasites, they leave the site of infection and they go into the spleen or the liver and they cause visceral leishmaniasis. All right, so if you actually look at the lesion, so this is a lesion, and you were to do a section through this, the lesions of these parasites are kind of interesting. So take a look at this lesion. What you see here, there are these holes. It almost looks like Swiss cheese, where you see these kind of big holes. These holes are actually vacuoles. These holes are vacuoles that the parasite has helped create. And so these parasites are actually living in these vacuoles within cells. I showed a little bit of it in this previous slide. So these vacuoles have parasites in them, and that's essentially what you're seeing here. That's essentially what you're seeing here. So the interesting thing about these parasites, Leishmania amazonensis, is that they form this interesting underlying architecture. There are other parasites that also cause cutaneous leishmaniasis, but the architecture of the lesion is different. So each one of these parasites establishes a certain relationship with the host. And really, the studies in my laboratory focus on understanding how these vacuoles are formed and the role of these vacuoles in the progression of the infection. So that's really what I'm very much interested in. So the rest of the talk that I'll be sharing with you, I'll be looking a little bit more at vacuole formation and start to think about ways to disrupt this process. So to me, when I first saw this image, so for me this is an iconic image. This is essentially a blown up section of these infected hosts. So this is tissue, and you can clearly see these huge vacuoles with multiple parasites. So one cell can end up with easily 20 to 30 parasites. So from the time the parasite goes in, it resides in this compartment. The compartment just continues to grow, and the numbers of parasites in that compartment increases. They increase. Contrasted with another presentation. So this is a cell, this is a macrophage. Each one of these things is a parasite. But what's interesting here is that each parasite is in its own compartment. So two very different niches of parasites for these parasites. In one of them, they live in small segregated vacuoles. In another, they live in these communal vacuoles. So again, the title of my talk is understanding how these homes or these different niches are formed, and really trying to get a feel for what the underlying biology is that results in this kind of process. The way I think of this kind of project is, the more you know about how you get to here gives you insight into the steps in which you can probably disrupt the process. And if you are able to find the right step to disrupt the process, hopefully you'll be able to come up with an intervention, a drug, to limit the progression of the infection. So, and I just wanted to show you this again. This is a huge vacuole. This is one of those communal vacuoles. You see the parasites in here, and these are the individual cells, each one of them in its own vacuole. They're all different. So the biological question here is, where does this membrane come from? So when a parasite goes in, so clearly to make a membrane, if you keep on distending this compartment, you need to keep getting membrane from somewhere to essentially help in the growth of that, in growth of this vacuole. Same here. Each time a parasite divides and the daughter is going to separate vacuoles, membrane has to come from somewhere. So you can essentially look, if you set up your experiment, you can essentially try to follow the origin of membrane that results in this kind of presentation in the cell. All right? So to set up one's studies, it helps to have a hypothesis. It helps to have a hypothesis. Most of us have what's called hypothesis-driven research. So you essentially have to establish a hypothesis. When we started this work, there were certain things that we knew about these vacuoles. We knew that these vacuoles, there are molecules that these vacuoles had proteins that came from certain host cell compartments, like lysosomes. So we knew that the niche of these vacuoles were in part derived from lysosomes. What we didn't know was, and we still don't really know, is what the contribution of the parasite is. We want to know what the parasite makes to help induce the formation of vacuoles the way that you see. The other thing that I already started discussing with you is, we want to know where the membrane is coming from. Is the membrane coming from the lysosomes that are interacting with these parasites, with these vacuoles, or is the membrane coming from another source, like the endoplasmic reticulum? If that is the case, then we kind of want to know if there's a way that we can disrupt it. So our hypothesis is that the secretory pathway components, that essentially the membrane is coming from the secretory pathway, and that those components come there through vesicle-mediated traffic. I'm going to try and develop that a little bit. And so essentially, this is a scheme of how a parasite goes into a cell. So the parasite is big. It's taken through a process of phagocytosis. And the initial phagosome that's derived starts to have interactions with host vesicles or host organelles, like early endosomes, late endosomes, and lysosomes. So there's interaction there. So as the parasite's vacuole grows, the interactions continue. When we started these studies, there was understanding that these events took place. The secretory pathway is essentially what happens is a pathway that's used by proteins that are synthesized in a cell. So inside your cell, the site where your proteins are synthesized is the endoplasmic reticulum. From there, those proteins are processed through or they are taken through a series of compartments, the Golgi being one, and they undergo further processing. Essentially, those proteins are going to be proteins that are destined for secretion. So the majority of those proteins, and so that's called the secretory pathway. Our hypothesis is that proteins from this here, or membrane from the secretory pathway, those membranes are being recruited and they're helping in the development of this vacuole. That's our hypothesis. And that's what we have essentially, what my lab has tested. So how would one go about testing that kind of hypothesis? One thing that we could do is, if we had a way of introducing proteins into the secretory pathway, we can then look to see if the protein that we introduced into the secretory pathway ends up into in the parasitophorous vacuole. So if we can introduce things into this pathway, demonstration that they end up in the vacuole will be supportive of our hypothesis that indeed this pathway interacts with this vacuolar compartment. And the way we did this is there are toxins. You might have heard of them. Ricin is a toxin that is it's a plant toxin. And it's actually a toxin that is that can be used as a bioweapon. These are interesting toxins. The way these toxins get into our cells is of interest. They actually get into our cells by essentially going through the secretory pathway in reverse. So the secretory pathway, as I showed you here, usually originates from the endoplasmic reticulum, and the proteins go in that direction. But there are certain things that can sometimes use that pathway and come in reverse. That's the retrograde pathway. So we took ricin that goes down through this retrograde pathway and asked whether ricin is going to end up in our parasitophorous vacuole. Because if it does, it would be a demonstration that molecules in this pathway can end up in parasitophorous vacuole. So the good thing about this all is that we can tag ricin. We can actually purchase ricin that has a fluorescent tag. And so we can follow the progress of ricin in a cell. So the way ricin, the reason why ricin is toxic is that when it goes through the secretory pathway, it ends up in the endoplasmic reticulum, and then from the endoplasmic reticulum, it blocks protein synthesis. So when ricin goes into a cell, it blocks protein synthesis and the cell dies. So if you ingest ricin, essentially ricin causes all of your cells to stop functioning, and then you die, right? So that's how ricin works. And so we thought if we can follow the progress of ricin in a cell, we could look to see whether that ricin ends up in a parasitophorous vacuole. So this is kind of what this is the way the experiment looks. So here's a cell. The ricin is tagged with green. So we're looking at this under the microscope. In a fluorescence microscope, the ricin is tagged with green. Within a few minutes, we can see that ricin is at the periphery of the cell. And as we go down in time, ricin starts to go into the cell, and and essentially by 3 hours, when you what you see here is that ricin is actually getting out of the ER and it's in the cytosol. So we can actually monitor the progress of ricin, and at different points we can say, okay, ricin is now at the Golgi, at the level of the Golgi, or we could say ricin is now at the level of the endoplasmic reticulum. And so we can trace ricin, and hopefully we'll be able to determine whether ricin has made it into a parasitophorous vacuole. So here is essentially another look at this, but now we have a cell that is infected. So here is the nucleus of the host cell. It has a parasite that lives in a vacuole there. So when we expose the infected cell to ricin, we see at a very early time point that ricin is at the membrane. And as we go on in time, we now start to see that ricin, which is in green, ends up inside the vacuole. So our vacuole here is labeled with LAMP. LAMP is a lysosome molecule, but that's the compartment that the parasite is in. And essentially, over time, we see that ricin ends up in the parasitophorous vacuole, right? And essentially, that kind of study will confirm to us that indeed ricin is trafficking. When we introduce ricin and ricin comes in through the retrograde pathway, it ends up in the parasitophorous vacuole. So one of the things that's interesting about this kind of study is we can look at this and we can monitor the rate of accumulation of ricin in these parasitophorous vacuoles. And what we saw was that these ricin accumulated slowly. It was a progressive accumulation. And this is kind of an important thing, because if the ricin, all of the ricin that you put ended up all at the same time in the vacuole, it would have suggested to us that there is probably some kind of quick fusion that occurs. But instead, what we saw was a slow increase. And what we determined was that that slow increase was probably because vesicles were going from one compartment into another, and that transfer of ricin was a vesicle-mediated process. Now, the good thing here is there are drugs that we use to block ricin. So brafaldin A, for example, there are drugs that we use to block ricin. So it allowed us to confirm that we're actually looking at ricin that was in this retrograde, going through this retrograde traffic. So what that allowed us to do, to conclude, is that indeed the secretory pathway interacts with the parasitophorous vacuole, and that there are components of the secretory pathway that are important for the building of the parasitophorous vacuole. So if you now go back to this figure, remember I showed you that there were these arrows that showed that there was interactions. There were interactions between the endocytic pathway with a parasitophorous vacuole. Now we know that the secretory pathway, there's actually vesicles that are going into the parasitophorous vacuole. And so we at least have a good result here where we showed for the first time that secretory pathway molecules end up in the parasitophorous vacuole. So that observation is good, but the next question that we wanted to know is, what would happen if we disrupted that process? What if we blocked these interactions from occurring? Would we change the vacuoles, the parasitophorous vacuole development scheme? Would we change, would we be able to interrupt the replication of the parasites? So our next question was to figure out how to block that interaction. And it was important that we had determined that vesicles, that these proteins were going by vesicle transport. And so we could think of ways to block vesicles that originate from the ER from interacting with the parasitophorous vacuole.
Kima, a question. So the vesicles that are contributing to the PV, they come from the ER, or do they go back to ER, Golgi, PV?
So the question that you're asking is a good one. So the question is, what is the true path of those vesicles? We don't know the complete path of the vesicles. We know that there are ER proteins that are ending up here, and we kind of know that there are Golgi proteins that are ending up there. So the way we would, and this is work that is ongoing, we would actually need to be able to trace each one of these things to really figure out what is the actual path of each of these proteins. But the likelihood is that there is a lot more traffic. So it's not only do you have traffic from here, you have traffic from other parts of the secretory pathway. All right, so that is the take home, and those are experiments that are ongoing. So what I'm going to show you, and it will be brief and I won't go into any detail, is the fact that there are approaches that we decided to use to try and block this, this traffic. And this is a little bit of a complicated slide. All I want to show you is that for each of these vesicles, for trafficking of each of these vesicles, there are specific proteins that mediate that traffic. So if you think about it, in your cell, when you synthesize proteins, proteins have to be directed to different places. So there needs to be very tight coordination of vesicle traffic. It's almost like you going out on the street and there are street lights and there are cops directing traffic. There's a lot of proteins that are playing a role in where vesicles go to and which compartments those vesicles interact with. So if you found out what some of the key proteins are that mediate those interactions, you can figure out how to block them. And that's essentially what this figure is showing, that there are these proteins that are called snares that the question that we're asking is, if we blocked those vesicles, how would it affect the parasitophorous vacuole? Would it change the biology of the parasitophorous vacuole? The simple, so there are different strategies that we used to disrupt those proteins and disrupt those vesicles. What I just kind of want to show you is one of them. There are a couple of them. So this is a typical, these are vacuoles. So wild type, this is normally how big the vacuoles are. In this particular experiment, we disrupted one of those proteins through by using what are called dominant negatives. But the bottom line is, you see that this vacuole is gone from being big to being small, and there are these small vacuoles. So essentially, what it's suggesting is that when we disrupt that traffic from the endoplasmic reticulum or from these vesicles, we're essentially disrupting the development of this parasitophorous vacuole. But not only are we disrupting the development of the parasitophorous vacuole, it turns out that we're also disrupting the replication of the parasite inside. So, essentially, that's what these things show. So by using different methods, we can go from large vacuoles to small vacuoles. Yes?
In the PV, once they're making it smaller, does that decrease the number of the parasites inside the vacuole?
Yeah, so that's a good question. So the question is, does shrinking the size of the vacuole result in fewer parasites? The answer is yes, that both things are happening, that you are shrinking the size of the vacuole. And essentially, that's kind of what we are showing here. We measure the size of the vacuole and we measure the number of parasites. And we've done it in many ways. But yeah, so that was what was intriguing to us, that not only were we shrinking the size of the vacuole, but we ended up with fewer parasites. So I'll just jump ahead, and one of the interesting things here is a group in France was also interested in ricin toxicity. So ricin is a problem, and this group in France was interested in ricin toxicity. So they wanted to figure out how to block cells, the toxicity of ricin. And what they did is they screened a lot of small molecules. You can generate a library of small molecules, and you can essentially set up a screen. And when they set up that screen, they identified a few small molecules that can block ricin toxicity. And so we wanted to know whether those molecules that they had identified will work in our system. So this is the small molecule that they identified, dihydroquinazolinone. They called it retro-2 because it blocked retrograde transport. So we've used this particular molecule, and I'll show you that in our system, so pretty much the same kind of setup. Here are, here's an infected macrophage. When you start adding increasing amounts of the compound, you end up with smaller parasitophorous vacuoles, and you end up with fewer parasites. So back to the question. So not only are you shrinking the vacuole, but you're ending up with few parasites. And the reality is, the only reason why it's one is because the parasites that the cells that are cured, you cannot count. But the bottom line is, this molecule can do pretty much what we thought it would do, which is reduce the size of the parasitophorous vacuole, and then block or kill the parasites that are there. So we've, in collaboration with a professor at Brown University, we've been able to test secondary analogues of, so the original small molecule, we now have secondary analogues that we've been testing. And we now have compounds that are performing even better than the original compound, but than the parent compound. So we've even tested it in mice, the original parent compound. So this is the progression of a lesion in a mouse, and the way we do this experiment is if you inject parasites into the foot of a mouse, that foot, essentially a lesion forms in that mouse. And so you can come back and test a drug by introducing the drug and seeing whether the lesion shrinks and whether the number of parasites in the lesion, the number of parasites are reduced. And so you have a drop in the lesion size and there's a drop in the number of parasites. This was done with the parent drug. So now we are in the process of trying to assess the effect of the subsequent secondary analogues of the drug to see if this improvement is going to be greater, because in the test that we've done in vitro, there has indeed been an effect. Okay, so the interesting thing about this compound is that, and why we're excited about it, is that it treats this retro-2 can affect other infections. There are other infections that are affected by this drug. And in part, it's because those other infections, they use the retrograde pathway to get into cells. And so this drug is actually able to limit other infections, some of them viruses. So we're interested in the fact that, so the good thing is somebody is going to develop this drug, and we're interested in seeing how it affects our parasite. I'll just conclude here by saying, it's essentially repeating what I've told you. LPVs acquire molecular components from the secretory pathway. I showed you that. The secretory pathway appears to be a source of membrane, and when we target the snares, when we target these molecules that mediate the fusion of vesicles, we're able to limit the infection. So I'll stop here and acknowledge some people. I actually did put Tina is on there because she is helping with some of the studies, not those ones that we've shown, but she's essentially going to be helping with these studies. It's important to appreciate that all of the work that I showed so far was work by graduate students. So the first graduate student who started this work was Blaise Njamen. Jonathan Canton continued the work. We've had some summer interns that have helped us. These were high school students who came in and helped out. So the studies in the lab are done by graduate students. Charles was an undergraduate student who was in the lab for about 2.5 years. This year he's in medical school down in the Caribbean. So efforts like this are done by not just graduate students, it's undergraduate students. There have been postdocs who've been here. I noticed that I don't have, what's her name on there, but she did not do, yeah, Shamiksha, she didn't do that work, but she certainly was a postdoc in the lab. And our collaborator at Brown University, who's been providing us with these compounds, is Dr. Jason Sello, and we've obtained funding for our research from the NIH. So that's pretty much what I wanted to discuss with you about my work. So I'll stop for a minute and ask if you guys have any specific questions or things that were not clear from the kinds of studies that we've been doing. Yes?
Does this parasite only affect mammals, or has it been found in other types?
So this parasite, so there's a whole family of parasites there. These parasites only affect mammals. But of course, there's the vector that transmits them. But yeah, they only affect mammals. So not only do they affect humans, they affect dogs, they affect, there are rabbits and stuff that are infected with this parasite in the wild. But yeah, this is a parasite that mostly affects humans. Horses, I mentioned the fact that there are horses that are affected. Yeah.
Since you've been testing on mice, you said that where the sand fly bites is where is the coagular lesion. Since they eating it instead of getting bite or like put it in a certain place, are the lesions there? I suppose they're intern, right? Are they, or they show up also like in another external part?
So I'm not sure, so in the case of, I'm not sure that I get the question fully. You're asking about the
Where does the lesion shows up in the mice?
Oh, okay. So in the mice, the lesion shows up where you put it, where you introduce the parasites. In our particular system, we introduce the parasites in the foot, the back of the hind foot. So you see a swelling or a lesion that develops there. There's another approach, putting the parasites in the ear of a mouse. So the reason why these parasites are somewhat temperature restricted. So for the parasites that cause cutaneous lesions, they work better on peripheral areas. So for a mouse, if you were to put it on the skin of a mouse, that parasite will not develop. But if you put it on the hind foot, on the tail, the parasite would develop. So temperature is a limiting, is one of the limiting things for the parasites that cause cutaneous lesions. For the parasites that live in the liver or the spleen, what you'll do there is you just inject the parasite into the blood. It will find its way into the liver or the spleen, and it will divide in there. So each of these parasites they each have slightly different kinds of behavior. Yeah.
Other than the retrotube, other than it shrinking the vacuoles, does it do anything else? Like, does it have any other side effects in the body?
So one of the interesting things that's exciting about this drug is that so far we've not been able to observe any toxicity. So the original group that developed this compound, they were able to introduce up to 400 milligrams per kilogram into a mouse. So it's almost like putting a lot of that compound in a mouse, and it caused no toxicity. We've done it too. We've been able to go up to about 150 milligrams per kilogram. So we cannot, for all the things that we look for as far as toxicity goes, we cannot find any toxic effects, which is one of the things that makes us quite excited about the compound, and that you can introduce a lot of that compound in a host, because really, even though a compound might work well in vitro, in a test tube, that compound is not going to be any good if it causes side effects inside a host. And the fact that this compound doesn't lead to any kind of side effects is something that's of great interest to us.
Thank you. So you can now tell us a little bit about
Yeah,
your story.
yeah, indeed. So I've been interested in infectious diseases actually for quite a while. My story is very much like many of you all's. I actually started college in a 2-year what are you guys in? In a community college. I actually started my schooling in a community college in Maryland. So you probably can tell that I'm not an American. I grew up in Cameroon. And so I grew up in a place that where a lot of infectious disease abound. So I've always grown up seeing a lot of the parasites that I lecture to discuss with my students are parasites or infections that I grew up seeing. So essentially, when I thought about college, I always thought, hey, I'll go to college, I'll go become a doctor. It's not until I got to this community college in Maryland, I took organic chemistry, and my organic chemistry professor was one of those guys who he used to ride me big time. He made me, but what it did is it made me study organic chemistry. And I started to realize that what really interested me is really basic interactions. What are the basic interactions that lead to this much more complex organisms that we are? All of us, inside of us now, you have small molecules that are having important effects on us. I mean, if you go back and you look at molecules like serotonin, these are small chemical compounds that you probably, if you take in organic chemistry, there are compounds that look like those. And it started to fascinate me to ask how those compounds are behaving in the context of biological systems. And so what I started realizing that was really of greatest interest to me was to ask questions, very basic questions. And so after my 2 years in community college in Maryland, I transferred to the University of Maryland, Baltimore County, and there I got a bachelor's degree in biochemistry and molecular biology. So that actually is where I started. And it's a challenge as you're going through all of these, each of those stages, trying to determine which way should I go. I mean, I probably, in retrospect, I could have gone to medical school, but I know that I knew that I was really interested in asking more basic questions. But what I actually did was I went to public health school. So I went to Tulane University and did public health. It gave me a broad view understanding of parasites and the communities that are affected by parasites. And after that, I figured, okay, now I got it. I know what I want. And I ended up going to do a Ph.D. And when I did my Ph.D., I studied how the immune response deals with Plasmodium or malaria. So for my PhD, I focused on malaria and how the host deals with that. I was interested in vaccine development and all. but at each point, you are always confronted with a question: which way should I go? I mean, do you follow? I mean, but you kind of always have to keep challenging yourself. Ask yourself, what am I really interested in? And figure out a way to pursue that path. So even though I studied malaria, there were some basic questions about the biology of how the parasite interacts with its host that were not really, I was not getting the right kind of answers that I wanted, so I changed systems and got into Leishmania. This parasite lives in a macrophage. There are certain kinds of things that we can do in this system that at the time I didn't think we could do with Plasmodium infections. But if you were going through that same path, you might end up with a different decision and go in a different path. But that's kind of the beauty of it all. In that, I mean, what you kind of have to keep doing is asking yourself, what am I interested in? And are there individuals or are there facilities in my environment that could help me get to where I want to go? I mean, that's the question that you continuously have to ask. But there are challenges, no doubt, as you go through and you ask those questions and you start to go down and actually do the work. There are clearly challenges all along the way. But if you are true to yourself, you will find that if you work hard, things will come.
So our time is up, but you can, if you want to approach him, you can talk to Kima.